Thank you everyone for coming on today. We've got quite a few online at the moment, and this is the third session of the 2023 Spring Series. Now, since the foundation was established in 2018, we've curated and sponsored a series of research programs into quantum biology and bioenergetics. And I'm delighted in this series that we're talking about the work that we've been sponsoring and supporting from our principal investigators. Now, in the first session, Rhys Mould, who's on the call today from the University of Westminster, presented the findings on the role of biophotonics and electromagnetic fields in cellular communications and bioenergetics. And in the second session, Stan Botchway is on today, and Alistair Mackenzie, I think Alistair is not on yet, presented on the development, visualization, and modulation of biophotons in living systems. Now, the first session video is now available on our website and the YouTube channel, but we have blurred out one or two of the slides pending the submission of the Rhys's publication. And in terms of the second session, the whole video will be uploaded once Stan and Alistair have submitted for publication. So in today's session, I'm really delighted that Philip Kurian, founding director of the Quantum Biology Laboratory at Howard University, one of our long-standing advisors, is presenting on the cooperative and coherent quantum phenomena in the life sciences. Now, Philip's been working with us on this project since 2020, and I'm going to ask Betony to give an introduction in a moment. Betony Adams works for the Guy Foundation and really is in charge of our scientific communications. She comes out of the University of KwaZulu-Natal in South Africa. And Betony is going to introduce our speaker, very well known to many of you, Philip Kurian, today.
Thank you so much, Jeffrey. It gives me a very great pleasure to introduce Dr. Philip Kurian as today's speaker. He will be speaking about his research, cooperative and coherent quantum phenomena in the life sciences. Dr. Philip Kurian is a theoretical physicist, researching scientist, and essayist, serving as a principal investigator and founding director of the Quantum Biology Laboratory at Howard University. He began his career as a maths teacher in North Philadelphia, and then completed his doctorate in physics at Howard after a stint at NASA Goddard Space Flight Center. Dr. Kurian is now the recipient of fellowships, grants, and awards from the Alfred P. Sloan Foundation, the US-Italy Fulbright Commission, the Whole Genome Science Foundation, the National Science Foundation, and the National Institutes of Health. His lab studies how collective and cooperative quantum behaviors can explain biological phenomena at the mesoscopic, organismal, and clinical scales, including in neurodegeneration, cancer, and even human consciousness. In 2020, the Quantum Biolab became the first group in the US and the third in the world to receive a scientific grant from the UK-based Guy Foundation, and lab's expertise is solicited regularly by federal agencies, national academies, and private foundations. In 2021, Dr. Kurian was appointed as a lead expert and organizer for the National Academies of Sciences, Engineering, and Medicine workshop on quantum-enabled sensing and imaging for biology. In 2022, he became a Simons Scholar and Senior Fellow at the UCLA Institute for Pure and Applied Mathematics, where he developed advanced theory and methods to understand complex, open, and excited quantum systems. Dr. Kurian also serves as a scientific advisor to the Science for Seminaries program of the AAAS Dialogue on Science, Ethics, and Religion, which seeks to further integrate frontier science questions into conversations among future theologians and clergy. His essays on science, human knowledge systems, empire, and spirituality have appeared in various media outlets, including the Los Angeles Review of Books, Granta, and Plough. So if you've attended any of the Guy Foundation lectures so far, you will be well aware of the fact that light plays a central role in the Foundation's research interests. In the first two lectures of the series, we heard various presentations about the endogenous production of light in living systems. Philip's work builds on this by looking more explicitly at novel quantum effects that might be taking place in biological systems when they interact with light. Light is central to quantum mechanics, which to a large degree was developed as a response to light-matter interactions such as blackbody radiation and the photoelectric effect that could not be explained using classical theories. Quantum biology, by extension, might be considered to be at least partly an investigation of light-matter interactions in where the matter is living matter. Some of the founding figures of quantum mechanics were already interested in how the new theory might offer insights into the ultimate mystery of what exactly life is. Perhaps even more relevant to the Foundation's therapeutic interests, Niels Bohr, who was famous for applying quantum mechanics to the model of the atom, gave a presentation entitled Light and Life at a convention that was specifically targeted towards light therapy. So if quantum mechanics looks at the interaction of light with matter and quantum biology looks at the interaction of light with living matter, then a good understanding of what exactly this matter is seems essential. And as any of you who have heard Alistair's presentations knows, the Gaia Foundation's interest in this direction was sparked off by their interest first in cannabinoids and then more generally in plant compounds that interact with light. Chromophores are light-sensitive molecules that are found in biological materials and are central to a number of research areas in quantum biology. This is most obvious in the context of photosynthesis, where chlorophyll is the chromophore that plants use to harvest light to create ATP. But the Gaia Foundation also has an interest in other contexts in which these light-sensitive molecules play a role, even, as I'm sure you have heard Alistair discuss, being implicated in the origins of life. And today we will hear about one chromophore in particular, which is called tryptophan. So tryptophan is found in many different contexts in biological materials, but there's one in particular that has attracted quite a bit of attention in quantum biology. Tubulin is a tryptophan-rich protein that is essential in the formation of microtubules, which are cytoskeletal filaments that play an essential role in a number of biological functions, including giving cells shape and structure, enabling transport of molecules, as well as mediating the separation of chromosomes in cell division. Microtubules feature in a number of contexts in quantum biological research, including theories of consciousness. What makes them so interesting and so amenable to hypothesis is both the light sensitivity that tryptophan lends them, as well as the particular networks in which these tryptophans are arranged, which you will hear a lot more about today. For the Gaia Foundation, microtubules are additionally interesting because of their important interactions with another focus of interest, which is mitochondria. And finally, why are networks of tryptophans so interesting? And you'll hear a lot more about this from Philip, but very briefly, how does these networks allow for novel quantum effects as compared to the sorts of light-matter interactions we heard about in the first two lectures of the series? As I've already mentioned, one of the core concepts of quantum biology is the interaction of light with matter, but what does this interaction actually mean? So very basically, light can transfer its energy to the electrons in a material, causing them to occupy a higher energy or excited state. And the simplest form of this can be modeled as a two-level system. There's a lower and a higher energy state that is coupled to a specific energy of light. In the simplest case, light will interact with only one of these two-level systems, a sort of one photon, one electron interaction, but in special circumstances, light can couple collectively to more than one two-level system at a time. And I will leave it to Dr. Kurian to elaborate on the advantages and implications of these interactions. Thank you.
Thank you so much, Betony, for that wonderful and detailed introduction. Hearing and reflecting and preparing for this talk, I reflected a lot, not only on the period of the grant, which has started from 2020, but also the last decade, because these insights we had been thinking about from 2013, 2014, but the people that we've met along this journey that have enabled and kind of catapulted the work out into the forefront have really been integral at every stage. So whether it was a conference in Helsinki in 2015, where I met Giuseppe Vitiello, or in 2016, where in Durban, I met my longstanding collaborator, Luca Celardo, and then in 2017, meeting Maja Chergui in Switzerland, and you'll hear about how that work plays into our collaboration. But then, of course, in 2018, I met in Vilnius, Jeffrey and Alistair, and that the conversation that emerged from that dinner in 2018 then led to conversations with Stan and Jimmy and Wayne and others on the call and Nina, at Cheddington in 2019, I believe. So it's as each stage, as we've moved toward putting the pieces together with the right people, the right skills, and the right theory, to then be at this place where we can now really see what the last 3 years have done in terms of harnessing the right experimental data to prove these predictions that we've had for almost a decade is really very satisfying. But I hope what I can do today is convince you a little bit about the magnitude and import of, as Betony said, why these collective interactions in biology are so robust and potentially transformative. So I just told you a little bit about my road to quantum biology, but disciplinarily, my road has come through theoretical physics, which, physicists are very fond of simple things that are exactly solvable. And so the two-level system, treating a ground state and excited state, we go to town with treating matter in this way. Now, of course, matter is not a two-level system. It's not a harmonic oscillator, but these things are nice because they actually give predictions that match with a lot of the experimental data we see in the universe. And so even though reality is not physics and physics is not reality, I want to remind everyone of John Archibald Wheeler's nice little essay, Law Without Law, that he wrote 40 years ago. And he has this picture of reality here, which is an R. And you can think of it as it's really what we're doing, whether it's in the Sky Foundation community or in science. We have a few clear posts of observation. They are not all the observations in the universe, but there are a few, right? And even within our mental schema and our constructs, we develop theories, what John Archibald Wheeler calls a papier-mâché construction of imagination and theory. And sometimes there are multiple ways to fit between those posts of observation a theory or set of theories that interpose between what we experimentally observe as science. Now, I found out actually at the GRC that there are not only spherical cows, but there are spherical birds. And as these migratory birds that we know of have taught us a lot about the orientation of living systems to the Earth's magnetic field, life maybe has a lot to teach us about the interaction of living matter with light. And the challenge here is that life is messy, right? It's not simple. Even though, theoretical physics is neither simple either, but these words are used to describe analogies for systems that are not exactly solvable, the nonlinear, non-equilibrium, and non-adiabatic systems, right? And so you have, for those who don't know, non-adiabatic is beyond the Born-Oppenheimer approximation, where electrons and nuclei are coupled strongly, usually due to the interaction with some type of photonic excitation. And so how is it that we have this long-range spatio-temporal order in biology, where two objects, an enzyme and a substrate, can meet at just the right time, at just the right spatial configuration, in just the right sequence, all according to what for many decades was determined to be strictly Brownian motion? It's a puzzle, right? And as we go from asking questions about two-level systems being excited by single photons, it raises additional questions about the quantum vacuum. And not this vacuum in a field theoretic context, but this vacuum. So if you think of the atom as a static entity, an entity that exists in and of itself with calm orbits, it's nothing like that, right? It's a vast sea of photons coming into and out of existence, virtual and real. And those photons, this background of light that emerges and goes back to the quantum vacuum, is what we use as the building blocks to build life. So between this atom that I showed here in this video and what we see in the bearded reedling, we have at least 18 orders of magnitude in space and time. So across those 18 orders of magnitude, why are there so many roads to quantum biology? If you're not a physicist, that's okay, because we have to learn to speak across these many cycles, these many time and length scales, and there's new phenomena that arise as we see the interactions across these divergent length scales. So connecting them meaningfully in a way that speaks to, let's say, the theoretical physicist, the chemical physicist, the biological physicist, but then to the molecular biologist, the clinician. These are real challenges and something that the diversity of this community will speak to. So today, rather than speaking so broadly, what I will do is focus in on the work that we've completed over the last 3 years, specifically looking at ultraviolet superradiance in these mega networks of tryptophan in biological architectures, which are rather ubiquitous and found in many diverse biological systems. And in this work, I want to highlight the work of 2 of my postdocs, Nathan Babcock and Gustavo Montes Cabrera, and our collaborators, Katrin Oberhofer and Majed Churgui at EPFL, and Luca Cilardo at the University of Florence. So this work, if you're interested, is currently posted on the archive, and so any details that I miss here, you can go to the reference. So, as I said, these mega networks of tryptophan, which is a strongly fluorescent reporter, are ubiquitous in biology. And the key to tryptophan being a strongly fluorescent reporter is that it absorbs strongly in the ultraviolet, and then it emits also in the ultraviolet at a longer wavelength. And because it is so strongly fluorescent, it's been used to look at dynamics of enzymes. It's been used to look at a reporter of the Stark effect close to the lipid bilayer, to strong electromagnetic fields and electrostatic fields in the cell. So in this way, it's very sensitive to local and even inhomogeneous environments. And so different populations of tryptophan in proteins can actually be used as a marker for what is happening electrodynamically across many different regions of the cell. And so even though here you see in the panels below a number of really beautiful microtubule architectures that are based on tubulin, these mega networks of tryptophan are by no means limited to microtubules and those kinds of proteins. There's even a photoreceptor complex called UVR8 that is found in plants that actually uses a very intricate arrangement of tryptophans to funnel UV light energy into photoprotection, a photoprotection mechanism for plants. So as Betony kind of outlined, we are looking for collective interactions. Why? Because collective interactions are a key to the robustness of biology and to the hierarchical self-organization. What I hope to convince you of and present to you today is also how this collectivity can actually enhance quantum effects in a thermally turbulent aqueous environment, which is a key point to raise as we're thinking of quantum biology. But the simple picture I want to have in your head goes back to the 2-level systems. So if you can treat all that molecular complexity as a 2-level system, you have the picture that you see here on the left, right? Where you have a molecular resonance. So we can treat it as a 0 and 1 ground and excited state. And then you have an identical molecule on the right. Now, these molecules can couple into aggregate states, and we can actually see spectroscopic signatures of that. So on the right, here's an absorption spectrum where the center frequency, the 0 to 1 transition, is represented by this large peak in the center. And then the coupling of these two can produce an upper state and a lower aggregate state. So in different contexts, you might hear this as superradiant, subradiant, bright and dark, upper polaritonic, lower polaritonic in a cavity sense. But in either case, the coupling of the molecules produces a red shifted and a blue shifted state which we can identify in many cases as superradiant and subradiant based on their decay or spontaneous emission features. So how they emit light to the electromagnetic field. Now, what if we don't just have two systems of two-level emitters, but we have a whole network of two-level emitters? And that's the problem before us in these large architectures of tryptophan, right? So thanks to quantum mechanics, we can actually treat these systems of two-level emitters as a superposition state, right? So if even I have a single photon in this system, rather than exciting a single molecule, a single molecular resonance, I can imagine that single photon distributed in a coherent superposition across many of these chromophores. So you can see here in the notation I've written, can you see my cursor okay? Okay, so you see the first molecule is in the excited state, all others in the ground state. The second molecule in the excited state, all others in the ground state, and so on, right? And in quantum mechanics, we can create a superposition of these qubit states to produce a coherent superposition. Now, what's interesting about the phenomenon of superradiance is that even in this single excitation limit, we can have observable features in the decay rate. So as your system size scales with the number of molecules n, the spontaneous emission rate increases as n little gamma, where gamma is the decay rate of a single molecule or chromophore. And this has been explored in many works, but I highlight two here from Marlon Scully's group, which he calls this single excitation superradiance the super, what essentially puts the truly quantum features of superradiance at the forefront, and what we'll be exploring in this presentation. So here's the formalism, the mathematical formalism that we use, and it's a non-Hermitian open quantum systems formalism. And so for those of you who aren't physicists, open quantum systems are open to the electromagnetic field, as all living systems on Earth are. And so we have these quantum systems that are open to the electromagnetic field, and they evolve according to non-unitary dynamics. So this takes some getting used to, even for some physicists, because Hermitian physics is what normally describes quantum systems. And non-Hermitian physics allows us to then treat this interaction and decay to the electromagnetic field. So if we have a system of two-level emitters, you can imagine here the notation is I have on-diagonal elements that are described by NN. So if you think of a matrix, the on-diagonal elements are the NN elements and the off-diagonal elements are the MN elements. And so here, omega naught is that center frequency of tryptophan, and this little gamma is the single tryptophan spontaneous decay rate. Here now is a little more complicated. We have a coupling terms that arise from the interaction with the electromagnetic field. So because the electromagnetic field synchronizes behavior across all of these chromophores, that interaction with the electromagnetic field arises in this coupling. I won't go into the details, but using various sets of approximations, including the Born, Markov, and rotating wave approximations, we can derive these coupling terms for these systems of two-level emitters. And so you'll notice here that they do have imaginary parts, right? And those imaginary parts emerge because of the non-Hermitian nature of our interaction with the electromagnetic field. So if I were to just treat these two-level emitters as dipoles, because they're two-level systems, I can treat them as transition dipoles, I would get interactions that scale as 1 over r cubed in the coupling, 1 over r6 in the energy. So proportional to the traditional Forster coupling. However, because you have a system interacting with the electromagnetic field, you get for large R these long-range couplings that include 1 over r, 1 over r squared terms. And so you can think of it, even if you aren't familiar with the mathematical details, as the field essentially synchronizing the behavior of these two-level emitters. So you have this system, you can imagine a wavelength of light in a volume of lambda cubed. You have many of these two-level emitters, and in that volume lambda cubed, you have some level of synchronization and orientation of those dipoles to the electric field vector of the light. Now, the upshot of this kind of Hamiltonian is that you have complex valued eigenspectra. So what does that mean? Complex valued means you have a real part, Ej, and an imaginary part defined by this big gamma over J. You can think of them as the corresponding collective state parameters that correspond to the single tryptophan parameters omega naught and little gamma, okay? And so here we are going to be able to define these eigenspectra for any arbitrary system of these tryptophan networks. Now, in 2019, when we started these conversations with the Guy Foundation, this is where we began with our New Journal of Physics article. And so what we were interested in is this unique arrangement of tryptophans, ordered arrangement, in these microtubule structures. And it's not the best picture, but you can see here that those transition dipoles are oriented according to the position and placement of each of these tryptophan molecules. Now, a priori, it wasn't entirely clear that this was going to produce anything significant. But what we were interested in as we began building these structures was this observation that as we go from a single spiral in the microtubule to 10 spirals to 100 spirals, which is on the order of 800 nanometers, we see the emergence of this superradiant state close to the lowest excitonic state. And this was really intriguing because, of course, you don't see it at one spiral. And neither do you see it if we were to randomize the orientations of the dipoles. So we keep all of them in the same place, but we randomize the orientation. You can see in the lower right panel D that all the superradiance goes back to where you were at one spiral or less, right? But hundreds of times the single tryptophan decay rate is significant and observable and measurable. So if you want a mental picture of how a superradiant state looks in a quantum sense, here we've plotted the probability amplitude, or actually the probability of that superradiant state being distributed on the set of 10,000 chromophores in this 800 nanometer microtubule. And so you can see here, the majority of the probability distribution actually situates on the outer surface. Now, if you go to the reference, you'll also see we've plotted a subradiant state, which I'll go into more detail later. And these subradiant states are actually located on the inner lumen surface, the majority of their probability distribution. So even though they are dark, these are long-lived states that exist much more on the inner surface, and that may have functional implications. So returning again to these microtubule structures that Nathan has done such a wonderful job building computationally, here we have a single tubulin dimer, and there are 4 tryptophans in the alpha and beta monomer, colored here in red and blue. There's 8 total. Here you have a microtubule spiral that is formed. There's 3 spirals here, as you can see. And then from those basic building blocks, you can build a number of different architectures that show up in the cell. Here in panel D, we have the axoneme, which is the structural core of many cilia and flagella that are used for locomotion and even in the lungs and even in the neurons for moving things along in the cytoplasm. You've got in the centriole this kind of vortex arrangement of triplet microtubules, which the centriole is one of the most strongly conserved structures in all of evolutionary biology. It also has been shown to orient the cell to a light stimulus within a 2 to 3 degree azimuthal range. It's also the microtubule organizing center, and these two the centrosomes in the cell provide centers for mitotic cell division. And then in panel C and F, we have these axon bundles that are prototypical in mammalian neurons. And these form large structures from 7, 19 bundles, all the way to hundreds of microtubules in these bundles. And they are usually stabilized by the protein tau that mediates interconnects between these microtubules. And so they're very stable over long times with regular spacings and orderings that keep them kind of locked in place in neurons in several types of axons. So what did we predict and what did we measure? Those are the main questions that I hope to convey in brief today. So these are our predictions using all this superradiant formalism that I showed on the previous slides at thermal equilibrium. So we're in a very conservative environment where we're not assuming anything particular about biology or out of equilibrium, but we're assuming a kind of robust, thermally turbulent environment. And we want to predict how does the quantum yield look at equilibrium. And the quantum yield, of course, is the amount of photons, in other words, that are emitted relative to those that are absorbed. You can also reframe it in terms of the radiative decay rate. But here, you can see this probability P of T in this function on the right-hand side of the equal sign, which is the probability that you have an excitation in your chromophore network. This probability on the left, P sub kt, defines for that particular network the probability that you have an energy eigenstate indexed by k at time t with energy E sub k. And for those of you who are familiar, this value z is the partition function in thermal equilibrium. So as I mentioned, the quantum yield is a ratio of photons emitted to photons absorbed, but it can also be reframed in terms of radiative and non-radiative decay rates as follows. It's the radiative decay rate over the sum of radiative and non-radiative decay rates. And in thermal equilibrium, you can take the thermal average of those. So keeping in mind we have a non-Hermitian and therefore non-Hermitian Hamiltonian, which gives us non-unitary dynamics, we have that the time rate of change of this probability is actually proportional to the radiative decay rate. And so that radiative decay rate defines how the probability leaks out to the electromagnetic field. Now, if you were to take the electromagnetic field and the matter system and combine them into one, you would of course have a conserved probability. But in this case, we're interested in how that excitation propagates in the matter. We can get this radiative decay rate from the imaginary part of our complex eigenvalues that I showed you on a few slides ago. And the real part of those complex eigenvalues you can see in the EJ here. So we have all the tools we need to then compute the quantum yield for arbitrary structures of several, even up to nearing 1 million chromophores in these large structures. So for a microtubule, you see from here from 1 to 10,000 chromophores, this is akin to an 800 nanometer microtubule. But you see here we have these kind of leveling off features, these sharp edges. So this is, as you build a tubulin, which has 8 tryptophans, you see the slow rise. As you build a spiral, you go from 10 to 100 spirals. But the real increase, the observable, measurable increase that you see in the quantum yield occurs as you build the microtubule out to 10,000 spirals and beyond. But you see this saturation, which is expected given that the excitation wavelength is around 280, 300 nanometers. So as you get to a few times that excitation wavelength, you're going to see a saturation of this effect. Now for the centriole, which is of course much larger and has a larger diameter as well as more tryptophans because you have 27 microtubules, you see the same type of behavior starting though out at almost 3,000 tryptophans, but gradually we don't even reach saturation at 100,000 tryptophans. So this is on the order of, I believe, about 300 nanometers. And so for a 91 microtubule bundle, in an axon, you can even see there's even less saturation. And I'll go into more detail about showing, what does the superradiant eigenspectra look like for these large structures and how does that saturation actually play a role in enhancing the quantum yield. So I'll give you the conclusion up front is that our measurements of quantum yield with our collaborators at EPFL agree with our predictions at thermal equilibrium. So I want to walk you through a little bit of how we came to that conclusion. So here you can see in the upper left panel, the absorption spectrum for tryptophan, tubulin, and microtubules shown here in solid. And you can see that there's similar absorption maxima for all three, but the absorption emission Stokes shift, how much room you see here shifting from absorption to fluorescence in dotted, that is similar but much smaller for microtubules and tubulin. So this essentially means that the amount of energy, because tryptophan is more exposed to the solvent environment, its strong transition dipole reorients due to the water dipoles that are surrounding it. So it relaxes much more and then emits a lower frequency photon than a similar absorption emission event that occurs within a protein. So the protein within tubulin and microtubules, it emits, but at a higher energy than for tryptophan. Now, to compute the quantum yield, we have to take the quantum yield of a sample and the quantum yield of a reference. And the reference sample is, of course, tryptophan because it's so well characterized and has been for decades. And so here, F sub S is the integrated fluorescence area under the curve for the sample and the reference. Here's the absorption for the reference and the sample, and the index of refraction squared for the sample and the reference. And so these quantum yields were determined at both 280 nanometers, where many amino acids absorb, the aromatics and cysteine and histidine a little bit, and then at 295 nanometers, where only tryptophan absorbs. So we have two benchmarks to determining the quantum yield, and thankfully they both agree qualitatively, and that agrees with our prediction. So the scattering background of the microtubule solution was, of course, removed. But then we paid careful attention to the Rayleigh-like scattering that also emerged, or the geometric scattering that emerged from these samples. And you can see here, if we don't correct for this, this is something that could lead to serious errors in the computation of the quantum yield. But you can see that Rayleigh-like 1/λ4 scattering that we've subtracted from our microtubule sample. And you can see that above 280 nanometers, it agrees very well with the tubulin. So we can get an upper limit using the corrected spectrum and a lower limit using the raw data, and then take the average of those. And so that's what I've shown here. And I want to draw your attention to those quantum yields in the lower right corner of the table. So notice as you go from tryptophan to tubulin, you get a decrease in the quantum yield. So it was a bit unexpected in the sense that if you would see a very modest increase due to superradiance as you go from tryptophan to tubulin, because there's 8 tryptophans organized in a single tubulin. So you would see some, if not all, maybe you wouldn't even see it because of the disorder in the local environment. But now, because we see a decrease, we can confirm that there's an increase in the non-radiative decay rate as you go from tryptophan in solution to tryptophan in protein. And that's key, okay? The increase in the non-radiative decay rate as you go to the protein environment, as we go from tryptophan, as we go from tubulin to microtubules now, we somehow overcome the non-radiative decay rate increase to produce up to 70% increase in the quantum yield. And at 295, we see about a 50% increase in the quantum yield. So this is extraordinary because it confirms our superradiant prediction that, in fact, we do see an enhanced radiative decay rate, and that this is not due to a decrease in the non-radiative decay rate, confirming our prediction of superradiance. Now, to get some more resolution on the time of these states, we resorted to ultra-fast time-resolved spectroscopy at EPFL with our collaborators. And this is femtosecond transient absorption spectroscopy, which has sub-100 femtosecond resolution. And so we see the following kinetics for microtubules and tryptophan and tubulin, shown here, tryptophan in orange and microtubules in blue and tubulin dimers in green. And so the basic principle of FSTA spectroscopy is that you pump in this case in the ultraviolet, you excite your sample of tryptophans, whether they're in protein or in solution, and then you do a broadband probe across many wavelengths such that you can get kinetic spectrum at 295 nanometers, 280 nanometers, 310, 325, we can get all sorts. But I wanted to tell a story here. So I'm looking at 295 and 310, where only tryptophan absorbs. So we can be confident that we're looking at really the collective dynamics of tryptophan. And so here, what we were very pleased to find was this very sharp initial decay for tubulin dimers and microtubules. You can see that by the slope of these curves at both 295 and 310. Followed by this very long time component that is even comparable or greater than tryptophan, which decays on a time scale of about 2 to 3 nanoseconds. So these ultra-fast time-resolved experiments, we measured out to about 1.5 nanoseconds. That's 1,500 picoseconds. And so here we're just showing the first 30 picoseconds of data. And so this is interesting because the superradiant states that we predicted are predicted on this time scale for microtubules, on the few picosecond time scale. So this agrees with our prediction of superradiance. And this long time scale behavior would agree with our predictions of subradiance, because our superradiant states, as I showed in the beginning, are always paired with these lower aggregate states that are subradiant, that are dark states, dark in the sense of not coupling strongly to the electromagnetic field, but being long-lived. Now, you might also wonder, where are the centrioles? Where are these things? So we actually attempted these experiments with centrioles and with microtubule bundles, but these experiments were complicated by sample purity requirements with the centriole and getting sufficient signal to noise with the microtubule bundling. We've since learned a great deal, discovering a protocol from the 90s that can help us with the microtubule bundling, but this will require further resources and experimentation. So the main puzzle that we're trying to work out right now, though, is why the microtubule curve is not as steep as the tubulin curve. So this energy is essentially going rapidly into the protein complexes. It's being emitted by the tryptophan network, and it's likely being absorbed by other molecules that are in the protein, but then are being other chromophores that are nearby to the microtubule. But why the microtubule would have less energy going quickly into the surrounding environment is something that we're still puzzling through. So in the absence of experimental data on our centrioles and microtubule bundles, we resort to our theory and our simulation. And so here you can see in centrioles, this construction of 27 microtubules, you can see similar behavior, but at a much grander scale. So here we have superradiant enhancements that are several thousands of times the single tryptophan decay rate, little gamma. And so this is at realistic length scales for centrioles that are between 250 and 500 nanometers and sometimes longer. And so these dots here you can see are our numerical predictions. These are pretty hefty calculations that sometimes take several weeks up to a month, but now we have these analytical fits that you can do on a laptop instantaneously. So again, these are several thousand times the single tryptophan decay rate for the whole structure in the absence of disorder. But what about in a disordered environment, right? So we don't have these kinds of ideal networks that are situated with the electromagnetic field. So what we did was we artificially added static disorder to our system by taking each of the tryptophans and adding from plus or minus W/2 in disorder and taking a random uniform distribution within that range. And so we started from W equals 1 inverse centimeter all the way to 1,000 inverse centimeters, noting that about 200 inverse centimeters is physiological temperature. And what we were surprised to find is as you go from 64 nanometers to 256, so the longer your centriole, the more chromophores, the larger your system size, the more disorder it takes to disrupt this quantum effect. So contrary to the conventional wisdom, which says, okay, you need small systems, you need local control, biological systems appear to be robust to static disorder. This prediction suggesting that even close to 200 inverse centimeters you have of disorder, you have an order of magnitude increase shown here by 20 times the single tryptophan decay rate for a static disorder strength of physiological temperature. So even at the temperatures of biology, you've got a measurable, observable superradiant effect that we can pick up in experiments in vitro. And this is suggestive of the way that biology isn't trying to go for full-scale, maybe, quantum computation, which I'll discuss at the end of the talk. But minor enhancements, even order of magnitude enhancements, can be profound in terms of photoprotective and photosignaling behavior. So why is the quantum yield as a metric or as a parameter so robust to disorder? The key to that, using the same phenomena that, or the same approach or protocol that we used in the previous slide, is to see that our superradiant states lie close to the lowest excitonic state in our manifold. You can think of it as a ground state. So as you go from, these aren't high-level populated states, but in terms of energy, they're low-lying states. And so in the absence of disorder, our superradiant state is sometimes the lowest excitonic state and often close to that state. But as you add disorder to your system or you increase the temperature, this superradiant dipole strength gets distributed within kBt of that lowest excitonic state. And because the quantum yield goes as this equation, where we have for small values of EJ, high probability of those states being weighted, you see that we strengthen the persistence of this observed enhancement even under ambient biological conditions because of the location of these states close to the lowest lying state. And so here, for both a microtubule and a centriole of realistic lengths, you can see that even at physiological temperature, it's almost identical to what you see in the quantum yield at 0 inverse centimeters. And even at 1,000 inverse centimeters, you still see a potentially measurable enhancement. So this is far beyond physiological disorder, and we can still see observable consequences of this quantum effect. Okay. And not only in centrioles, this same phenomena occurs in axonal microtubule bundles that I showed earlier. And so whether you're even less than on the sub-100 nanometer scale, we can see several thousands of times the single tryptophan decay rate already emerging. And so as you go to larger and larger structures, on the right, we see here these larger bundles of several hundred microtubules, several almost approaching a micron in diameter. You see up to 7,000 times the single tryptophan decay rate. So this would have enormous consequences for a collectively excited state in the brain. Now here, as I said before, these points here are from very detailed numerical simulations that take on the order of weeks, sometimes upwards of a month, but they can be diagonalized for these large matrices, several 100,000 times 100,000 matrix elements. But now, thanks to the work with Nathan and Gustavo, we have these nice analytic fits that we can do on a laptop relatively instantaneously that have only two free parameters: the number of microtubules in the bundle or in the centriole, and the well, in the centriole there this doesn't vary, or the length of the bundle. And so these other parameters are fixed. L0 is parameter of the length of a single spiral. You have lambda is the wavelength of the excitation, and D is the number of tryptophans in a tubulin dimer, and NS is the number of tubulins in a single spiral. So this is 2 parameters, and we have these nice analytic fits for a range of these structures. Now, as I said earlier, this is not specific to tubulin only. This superradiant phenomenon occurs in transmembrane proteins and in other cytoskeletal filaments like actin. So on the right, you can see these modest but still significant. For larger actin filament bundles, you'll see even larger enhancements. But these actin bundles also produce superradiant phenomenon. But as the actomyosin machinery is perhaps more locomotive, you can also, or mechanical in nature, you can see kind of a diversification of the portfolio and how life might use these different structures, not just for mechanical motion, but for a range of light sensing, light actuating behavior. On the left, I want to emphasize this point again with the ciliary axoneme. So here we have an idealized axoneme on the top and a realistic axoneme made of PDB doublets, these protein data bank doublets that Nathan found. And so you can see here, we have these modest increases, several hundred times the single tryptophan decay rate, that are less than centrioles and neuronal bundles, but still significant, which alludes again to the primary purpose of these structures is mechanical or locomotive, but there might also be a secondary light actuating behavior. So what is all of this simulation, our predictions, and our experiments that validate this for microtubules? What is this pointing to? Well, we had wonderful talks earlier in the series with Reese and Stan, suggesting the importance of biophotons in mitochondrial and other cellular metabolism. So here is a schematic of that. Mitochondria emit reactive oxygen species when they conduct oxidative metabolism, and these ROS emit biophotons, ultra-weak photon emissions, across the UV, visible, and infrared range. And so in our case, how are these UV ultra-weak photon emissions being emitted, and how are they being absorbed and re-emitted or re-signaled for photoprotection and photosignaling? That is the primary biological question of interest. And how might this type of mechanism be used for novel forms of information processing in the cell? So to go a little deeper into that question, I wanted to compare these quantum optical networks that I've described in cellular biology with state-of-the-art supercomputers. So I want to draw your attention to the timescales here. Again, for these superradiant states, we are on sub-picosecond to few picosecond timescales. So this is 10 to the minus 12th of a second or faster. So these are faster, certainly, than thermal relaxation. So you have signals that are moving, and certainly much faster than Hodgkin-Huxley neuronal signaling on the millisecond timescale. So you have these signals which require us then to update our computational bounds for what the brain or cellular networks can compute. So for example, the conventional approximation for how our 100 billion neurons in the human brain can compute is at kilohertz, this Hodgkin-Huxley kind of neuronal signaling, millisecond timescale, which gives us 10 to the 14th ops per second. But if we're operating at the picosecond timescale with signals that are much faster than thermal relaxation, we have 10 to the 23 ops per second, which is far beyond the exascale where supercomputers are state-of-the-art supercomputers are. And this points us to the reality that the human brain and cellular networks are not processing information with the same energy consumption requirements of classical digital computers. And this can be seen very straightforward. If we look at Frontier supercomputer at the exascale, It operates at about 20 megawatts of energy. For comparison, thanks to Marco, we've got 1,000 megawatts is a nuclear power plant, right? Thanks to Marco for giving me that data point. And then if we have the brain at almost 6 orders of magnitude more ops per second, operating at 6 orders of magnitude less energy, 20 megawatts, which is less power than a single light bulb. So how is life evolving and dynamically processing information using ATP and other sources to conduct such efficient computations? If these things exist, life has shown us that life will use all the physical mechanisms at its disposal to optimize behavior. And because this quantum phenomenon is observable and measurable in a physiological environment, why couldn't life be using these tools to optimize and enhance behavior? So I also want to draw your attention again to the timescales of these subradiant states. So even though these are not as easily measurable, they are measurable, but not as easily, these operate in some cases at the tens of seconds of time scale. So you have this quantum phenomenon that operates both very fast and at long time scales, okay? So you can imagine both effects for integrating a very fast signal at short time scales and then integrating that with something that persists on long time scales. And that's something that we need to think a lot more about measuring and also thinking of the implications. So let's go back to this model that I presented at the very beginning of the talk, this system of 2-level emitters coupled together in an architecture. Now, as I said, you can write that as a superposition, a coherent superposition of excited and ground states, but I want to write it in a slightly more suggestive way. Let's write it in binary, and let's think of them as qubits. So this is actually what's called in quantum computing a W state. And a W state has some very interesting properties, namely that if you remove one of these qubits, you still can maintain the entanglement between the remaining n-1 qubits, okay? Now, this is not the case for all Bell states or all entangled states. But it's interesting because in biology, wouldn't it be interesting if biology has taken the W state and used it because we need something dynamic, not static, not something that sits in an existing configuration, but something that can grow and process and degrade if needed, right? Now, what's interesting about the W state is we can compute what's known as the von Neumann entropy, the von Neumann entropy being the quantum analog of the classical Shannon entropy. And by constructing the density matrix of this W state, which is rho W, we find that the entropy, the von Neumann entropy of this state vanishes. Now, this is unique to quantum mechanics because if we take this pure state density matrix and we transform it to a mixed state diagonal density matrix, we increase the entropy. So as we actually erase the quantum coherence information in our off-diagonal elements, and we look at the mixed state behavior, we increase the entropy as we add the components. So if we think of them as a single excitation on the first and the second, we have that along the diagonal in our density matrix, we actually increase the entropy. Now, this is unique because if you think of a joint system and you have entropy of individual components, you would imagine you put the joint components together, you get more entropy. But in quantum mechanics, entanglement ensures that we actually can decrease the entropy. And decreasing the entropy is a key point for life. So I referenced a few slides ago the energy consumption rates of these quantum optical networks or biological systems in the brain. And so that brings us to questions of computing, which brings us to the seminal work of Landauer, who in his 1961 paper actually didn't quote a limit, but rather a lower bound on the dissipative cost of logically irreversible operations. You can think of an irreversible operation as a many-to-one operation, taking many states and reducing them or selecting a single state. You can think of it as an AND gate, for example. If I have I say a 1 and a 0, if they're not the same, 1 and the 0, I return a 0. If they are the same, I return a 1, for example. So I take two things and I go to 1. So that's many to 1. Now, if you're a fan of the Copenhagen interpretation of quantum mechanics, you'll know that a measurement event, or when you make a measurement in the Copenhagen interpretation, you collapse a superposed quantum state. So the collapse of the wave function is actually a many-to-one operation, where you take the superposition state and you collapse it into a single eigenstate. Now, there's some subtleties here, because in thermodynamics, you have many copies of a system, but in quantum mechanics, you may have many copies in theory, but you could just have a single system in coherent superposition. Now, under the assumptions of Landauer's 61 paper, where you're talking about irreversible operations, because there's a whole field of reversible computing or reversible quantum computing, where you can go backwards and forwards one-to-one. But under the shorthands of its assumptions, you can derive this lower bound at about 0.7 kBt. So that's the heat dissipated to the environment per bit of information erased. Now, we can show using the lateral and longitudinal tubulin bond strengths that the energy input required per tryptophan qubit, not tubulin, but per tryptophan qubit, these two-level systems in the microtubule, is between 0.6 kBt and twice kBt, okay? So this is actually lower than the Landauer limit and slightly above. But if you need a comparison point, recall that the energy of a DNA phosphodiester bond or of ATP GTP hydrolysis is about 10 kBt. So think about how biology is operating. Biology is operating in chunks of 10 kBt. So whether you're putting tubulin together into a microtubule, it's constructing this mechanical architecture in these chunks. But the information processing may be operating at the level of very close to the Landauer limit, or even within 10 times if you want to think of DNA as a computing element. Now, compare this to modern silicon devices, which dissipate about 1,000 to 1,000,000 times the Landauer bound when reset. And so when I talk about resetting, you can think of it as a tubulin leaving the microtubule, right? You're resetting, you're degrading the information. So there's a trade-off, of course, between energy efficiency and speed. And for digital computers, the Landauer limit is only approached for very slow erasure processes. Which it then raises the question, is all of life and even the universe, are these computing substrates? Are we part of one grand scheme of computation by a higher mind beyond the universe, right? And so, you know, there are other theorists who have done a lot of work in this area, Paul Davies and Seth Lloyd, looking at the universe and all of matter, observable matter, and potentially even dark matter as computing substrates. So if you think about where we're operating in living systems, or where this work has pointed us to looking at the terahertz or the sub-picosecond timescale for computing in these quantum optical networks, what about at the Planck scale, which is 10 to the 44th ops per second? Now, I'm not saying that's physically realizable or measurable or accessible, but it gives us the potential to think of the matter in the universe doing this kind of computation at a much faster scale and with many more operations per second over a much longer time period, approximately 14 billion years. So we've got, so this is something that has arisen in the quantum computation literature about thinking of not just two-level systems, and this is how physicists work. We start with a really nice model system, and then we build up. So these qubit systems, which are three-level systems, and then qubit systems, which are multi-level systems. And it's interesting because all biological chromophores are multi-level systems in reality. None of them are single two-level systems, and often they're much more well described, like GFP, as a four-level system. You can get really good physics from treating them as 3, 4, and multi-level systems. And so ions like calcium, other magnetically sensitive objects in biology, you can treat them as multi-level systems. And there are interesting features and trade-offs and benefits for quantum computing architectures that are built on these qtrit and qdit systems. So for tryptophan, you can see here what we've focused on in this seminar is the 1LA state of tryptophan, which is kind of the lowest lying excited state. But if you study the ultra-fast dynamics of tryptophan on the, let's say, tens of femtosecond timescale, if you excite tryptophan, it excites to the 1LB state, which then quickly relaxes in tens of femtoseconds to the 1LA state. So for the simulations we're doing, we can focus on the 1LA state, which is what's operative after 50 femtoseconds. But in reality, you have these interactions between the 1LA and the 1LB state. And because of the reorganization of that 1LA state due to the aqueous or protein environment, you have this serious line broadening of that 1LA state that you can see here across the UV. And what we focused on were the 0-0 transitions. So a 0-0 transition is in the absence of vibrations, but you can see how those vibrations broaden the spectrum of LA such that they are both red shifted and blue shifted from the 1LB transition peak, almost surrounding the 1LB peak. And so there's a lot of confusion in the literature about this, the nomenclature, as well as how to how LA and LB relax or relate to each other, whether you're in an aqueous environment or gas phase or methanol or other solvents. But with all those details aside, it provides a rich playground, not even including other chromophores, to think about how biology might be using forms of quantum information processing. So I want to summarize a lot of the work that we've done in a picture, and what the work of this community has done over the last few years and where we continue to do. So this is kind of where many people start when they think about biology. They think of Brownian motion and thermal equilibrium. They operate in the Born-Oppenheimer approximation. They assume everything is weakly coupled because water is a liquid and in the bulk seems to operate as individual molecules, but of course we know better. And then they assume that there's no memory in the system, that there are only strictly Markovian dynamics. And then also, you know, whether you're a molecular dynamicist or a clinician, there's a presumption of ergodicity and linear response, that the response out from our system is proportional to the response that we give to it. And then finally, we also assume that things operate according to the laws of physics, according to electrostatic interactions, that they're local, right? That things operate, you know, including van der Waals effects, which are fundamentally quantum, at the few nanometer range. And that in fact, that due to Debye screening in an ionic environment, that we don't have multipole interactions at long range. But what I'd like to propose is a different vision for biology that comes from the quantum world and that helps us with understanding not just quantum effects, but classical effects in new contexts, out of equilibrium environments, non-adiabatic environments, which are fundamentally quantum, environments where the environment is strongly coupled to the system and you can't really separate them. Systems where you have non-Markovian effects, and then non-linear, dissipative, and even non-ergodic systems that do not explore the full phase space, because we know that even in out of equilibrium environments, cytoplasm and cells do not explore the full phase space of what's possible. And so there's sort of a selection in the out of equilibrium context. And then work from some people on this call, including Marco, on long-range electrodynamic selectivity at the scale of microns and beyond. So we have this beautiful picture of using conventional physics in the sense of out of equilibrium, non-equilibrium thermodynamics, quantum mechanics, even classical electrodynamics, but using these tools to understand biology as or the puzzle of long-range spatiotemporal order and some of these intriguing information processing effects that I've highlighted in the brains and in cells. So if you're unfamiliar with the Quantum Biology Lab, or if it's your first time at one of these sessions, and even if you've been a long-standing joiner, I wanted to give kind of a summary of the Quantum Bio Lab's research areas outside of this specific work. And so, you know, we have quantum biology at the center, but as you can see here, we're a group situated at the nexus of biological and chemical physics, theoretical physics, and applied maths. And so we've got these overlaps, and many different people from diverse communities have spoken to these in different ways. But we've got quantum thermodynamics, terahertz and Raman spectroscopy, and electrodynamics and van der Waals effects that emerge from very fundamental issues in quantum field theory, quantum optics, non-Markovianity and sampling, but as well as looking at really foundational questions for biologists on DNA catalysis, protein photophysics, and molecular dynamics. So we have some even pure biologists, some genomicists, some immunologists on the call. And so we've started pursuing questions along the lines of how do these electrodynamic behaviors actually impact, for example, G protein coupled receptors that are ubiquitous in serotonin channels, which have intriguingly relationships to tryptophan, both in biochemical and biophysical structure. So many of you were at the Gordon Research Conference last month, and so this is just a sampling. These are the different posters and talks that were presented that represent a sampling of the topics in my group, and these are the wonderful people that make that work possible that you can see here. So I wanted to conclude with a quote from one of the quantum greats, Max Planck, whose name is born on the Planck's constant that you've seen in all these equations in my talk, who said that no science can rest its foundation on the dependability of single human individuals. And the moment we have made that statement, we have taken a step which puts us off the logical pathway of the positivist system. We have followed the call of common sense. And he goes on to say, we have accepted the hypothesis that sensory perceptions do not of themselves create the physical world around us, but rather they bring news of another world which lies outside of ours and is entirely independent of us. So if you're interested in exploring these various worlds and the philosophical issues that arise from these very technical issues at the intersection of computing and biosystems and a life and light, we're currently hiring for a postdoc and a research associate to study these superradiant and subradiant effects at the organismal scale in Physarum slime mold, as well as at the neurobiological scale. So please do contact the QBL if you're interested in applying for these positions. And here, of course, without the Guy Foundation, we couldn't have done any of this work. So thanks go to the entire team, Nina, Jeffrey, Alistair, the advisors, Betony. Thank you for really making this work something that we can really stand on now, experimentally and theoretically. And it's something that we can really look forward to what the implications will be, both as we tease out the biology and we go to the clinic. So thank you very much for your time and for your attention.